Hello viewers, today we will discuss about the Lac operon. An operon is the group of genes with a single promoter. That means that more than one gene that is transcribed under a single promoter. The genes in the operon encode proteins that allow the bacteria to use lactose as the source of energy. We will discuss the Lac operon of E. coli, Escherichia coli bacteria today. In bacteria like E. coli, multiple genes are transcribed over a single promoter that is known as operon. For example, let's consider an operon system over here. Here is a promoter for Lac I gene. That is, Lac I gene will be transcribed over here and it will produce an inhibitor. Then we have another promoter and an operator, and then group of genes. The group of genes that are transcribed by this promoter are Lac Z, Lac Y, and Lac A. So the three genes, number 1, 2, and 3, are transcribed by this single promoter. All these genes will transcribe to produce protein. The Lac Z will produce beta galactosidase. Lac Y will produce permease, and Lac A will produce trans-acetylase. First of all, we should know that why the Lac operon is working in the E. coli and when the Lac operon system is needed. There are two states of Lac operon. Either Lac operon is turned on or it is turned off. It depends upon the availability of sugar, that is lactose. So basically this operon works for the breakdown of lactose because the bacteria, the E. coli, is using the lactose as a source of energy. When the E. coli uses lactose as a source of energy, when the glucose concentration is low and the lactose concentration is high. If the lactose concentration is low, the Lac operon will be turned off. And if the concentration of lactose is on, that means only lactose is available as a source of energy for E. coli, it will turn on this Lac operon system for the breakdown of lactose. So this is the portion of the Lac operon that is needed for the breakdown of lactose or for the hydrolysis of the metabolism of lactose. When the lactose is in the media, the three genes are transcribed by this Lac operon model: the beta galactosidase, permease, and trans-acetylase. Beta galactosidase is used for the breakdown of lactose, and the permease enzyme is used for the intake of this lactose by the membrane of E. coli. And same is the role of the trans-acetylase. The exact role of trans-acetylase is still not clear, but anyhow it also helps in the uptake of lactose by the membrane of the E. coli. So when the Lac operon is on, that means there is lactose in the media or in the environment, so that bacteria consumes this lactose as a source of energy. And then what will happen? The operon model will be turned on to transcribe all the three genes that are involved in the metabolism of the lactose for the source of energy. But when there is no lactose in the media, so why would the bacteria need to turn on this Lac operon? In that case, the bacteria do not need the Lac operon and the Lac operon model is turned off. How this happens? This happens by this promoter, that is promoter for Lac I. Lac I is another gene under another promoter, that is Lac I, and it produces a suppressor or inhibitor protein. When this protein is expressed, it binds with this promoter. Actually, in the absence of lactose, in the low concentration of lactose, when the concentration of lactose is very less, the P-lag produces inhibitor, and this inhibitor binds with this promoter. This is the inhibitor, it binds with the promoter, and the lag operon model is turned off, and all these proteins are not transcribed. But how? The question is how, in the presence of lactose, that lag operon model is turned on. Actually, what happens when the media is full of lactose, means the concentration of lactose is not less, the lag operon model has to be turned off. For this, the lactose is chemically modified to allolactose, and this allolactose binds with this inhibitor and make conformational changes in this inhibitor so that it cannot bind with the promoter of the lag operon. So when the inhibitor does not bind with the promoter of the lag operon model is turned on. Two regulators turn on the operon on and off in response to lactose and glucose levels: the lag repressor and catabolic activator proteins. Number one, the lag repressor. The lag repressor acts as lactose sensor. It normally blocks the transcription of operon but stops acting as a repressor when the lactose is present. The lag repressor senses lactose indirectly through the presence of this allolactose. The other protein is the catabolite activator protein. It acts as glucose sensor. It activates transcription of the operon but only when the glucose levels are low. So the catabolite activator protein sends glucose indirectly through the hunger signal molecule. As we all know, the hunger signal molecule, that is cyclic AMP. So the lag operon is an operon or group of genes with a single promoter transcribed as a single messenger RNA. The genes in the operon encode proteins that allow the bacteria to use lactose as an energy source. E. coli bacteria can break down lactose, but it's not their favorite fuel. If glucose is around, they would much rather use that. Glucose requires fewer steps and less energy to break down than lactose. However, if lactose is the only sugar available, the E. coli will go right ahead and use it as a source of energy. To use lactose, the bacteria must express the lag operon genes, which encode the key enzymes for lactose uptake and metabolism. To be as efficient as possible, E. coli should express the lag operon only when two conditions are met. Number one, lactose is available. Number two, glucose is not available. How are levels of lactose and glucose detected, and how do changes in the levels affect the lag operon transcription? There are two regulatory proteins involved. Number one, the lag repressor that acts as a lactose sensor, and the other is the catabolic activator protein that acts as the glucose sensor. These proteins bind to the DNA of the lag operon and regulate the transcription based on the lactose and glucose level. The lag operon contains three genes, Lac Z, Y, and A. These genes are transcribed as a single messenger RNA under control of one promoter. Genes in the lag operon specify proteins that help the cell utilize the lactose. Lac Z encodes an enzyme that splits lactose into monosaccharides that can be fed into glycolysis. Similarly, Lac Y encodes a membrane-embedded transporter that helps bring lactose into the cell. In addition to three genes, the lag operon also contains a number of regulatory DNA sequences. These are the regions of DNA which are particular regulatory proteins can bind, controlling the transcription of operon. The promoter is a negative regulatory site bound by the lag repressor protein, this one. The operator overlaps the promoter, and when the lag repressor is bound, RNA polymerase cannot bind to the promoter and start transcription. The catabolic activating protein binding site is a positive regulatory site that is bound when catabolite activator protein. When CAP is bound to this site, it promotes transcription by helping RNA polymerase to bind to the promoter. In the next lecture, we will study how the lag operon is regulated by the presence of cyclic AMP, by the presence and absence of lactose, and how it affects the binding of repressor and activator proteins. Thank you.